Hello, in this video we will discuss about the oncogenetics. So oncogenetic is the mechanism of cancer and tumor suppressor gene we will discuss also, which that is lead to the suppress the oncogene, and oncogene is suppressed by the tumor suppressor gene, which that is activate when activate tumor suppressor gene. So the oncogene will be inhibited by the their product basically inhibited. So the cancer factor we will discuss first is the oncogene activation and the cancer suppressor gene inactivation. So the cancer suppressor gene will lead to control the oncogene and inactivate the product of the oncogene. So remember about that. And rapid growth of cells and cell division abnormally and epigenetic modification of genome without the alteration of the DNA sequence, and family history, pollution and diet, smoking, a viral infection, alcoholism and medicine is the important causes of the cancer, or basically carcinogenesis. That is the factor is known as the carcinogen, and produce that process is known as the carcinogenesis. So let's begin to understand. First of all, we should understand about the cell cycle, and we can able to understand the DNA damage, as well as we will understand about the oncogenetics. So let's begin to understand first of all the G0 phase of the cell. G0 phase, the quiescence phase, which that's basically the G0 phase. The cell is basically in the time of rest. When the rest will be breakdown is go into the G1 phase, is known as the growth 1 phase. After the growth 1 phase is go into the S phase. Before the going into the S phase, the G1 checkpoint. This G1 checkpoint is basically used for the checking of the growth of the cell, and is used for the before the synthesis of the DNA. After this checkpoint is the G2 phase is progress, and after the G2 phase the G2 checkpoint is used. After the G2 checkpoint basically is checking for the DNA is double and correct. If not correct, that means it lead to separate and apoptosis by the another gene is known as the tumor suppressor gene, which that is used lead to the apoptosis. So the tumor suppressor gene we will also understand. So here is the after the M phase is the basically mitotic phase. Before the going to into the M phase is the checkpoint is known as the M checkpoint for using and checking for the going enter into the mitotic phase. So the mitotic phase is contain a prophase, metaphase, anaphase, telophase, and karyokinesis mean the nuclear division, and cytokinesis mean the cytoplasmic division. So the basically is the prophase is the prophase is basically is the beginning to going into the replication, while in the metaphase is the basically near to the metaphase plate, which that is chromosome are present, and that means the anaphase is the separating and telophase is completely separate, and karyokinesis mean the nucleus will be separate, and cytokinesis mean. So here is the histone protein. Histone protein is basically 8 subunit of histone protein is the type of 4 type of histone protein is known as the 1 nucleosome, which lead to DNA wrapping around the histone protein, and this DNA is the double stranded made up of nitrogenous bases adenine, thymine, adenine, and guanine with the cytosine, and that is the anti-parallel 5 prime to 3 prime, while the other is the 3 prime to 5 prime, that is anti-parallel. And DNA I want to show the structure here, and here is the methylation. Methylation is basically the epigenetics part, which that means the methylation is epigenetics, the modification without changes of DNA pattern. So when methylation will silence the gene, silence means the gene will be not active anymore, so it means the suppressed gene, and that means the not play functional important. And acetylation means the acetylation is expression of gene. It means that DNA will be expressed and transcribed into the translation and synthesis protein, while on the other hand the point mutation is the deletion of nucleotides, and inversion or addition is the basically is the point mutation, because on the basis of the nucleotide. So remember about that. So let's begin to understand these all things is lead to the cause cancer. Why? Because inhibition and activation of the genes which that lead to the cause cancer. So the causes of cancer we will discuss here is the activation. Basically activation of cancer is the activation of oncogenesis or deactivation of oncogene suppressing gene, tumor suppressor gene basically. So here is I am drawing the DNA structure which that is showing that contain oncogenes. These oncogenes is lead to proliferate the cell division and lead to transcriptional factor which increase the cyclin and CDK, which that is the cell growth protein, and which that is lead to increase the cell division. So here is the DNA segment which contain oncogenes and suppressing genes, or RAS or MYC gene. So this RAS or MYC gene is the cancer causing gene. Why? Let's begin to understand in the next. So here is the tumor suppressing gene, which that is when inactivate, it means that oncogene will be activated. It means tumor suppressor gene will be inhibited, control the oncogene. So the tumor suppressor gene is known as the P53 and APC and BRCA2. CRR2. So it's two types. So the cyclin and CDK growth factor, which that is the driver of the cell cycle. So first of all we will learn about the cyclin and CDK. So here is the early G1 phase is used for the cyclin attachment with the CDK 4 and 6, which that is basically activated maturation promoting factor will be bind, and this MPF will lead to trigger the E2F and retinoblastoma protein, which that is bind the complexes. This complexes will be break down by this MPF. So this MPF is the retinoblastoma is released separate, and the E2F is the basically growth factor. This growth factor is lead to G1 phase, which that is before the G1 checkpoint. So before the G1 checkpoint, the progress will lead, and this progression of the G1 phase to S phase. But the before the S phase, the one more cyclin E and CDK2 bind to end of G1 phase, which that is lead to enter into the S phase of the cell cycle. After the going into the S phase of the cell cycle is the cyclin A and CDK1 and 2, which that is lead to S phase progression. After the S phase progression, that is the G2 checkpoint is also occur. And after the G2 checkpoint is the G2 phase is basically begin. Before the beginning of the G2 phase is the G2 phase progression by cyclin P and CDK1. And after this basically is the here is the if the cell is not correct and damaged DNA will be separate abnormal cell. So this abnormal cell is tumor suppressor gene will lead to apoptosis and become a cell death. If the tumor suppressor gene is not activated, so inactivate by a methylation is known as the epigenetic will lead to the progression of the tumor and the proliferation of the cell division abnormally. So remember about that the CDK and cyclin is the driver of cell cycle. If low amount of CDK or cyclin will not allow to progress cell cycle. If high amount of CDK or cyclin will lead to uncontrolled cell division, lead to cancer. So we should remain constant and balance the cell division with respect to the cyclin and CDK concentration. So remember about that when the CDK and cyclin will be lead to increase due to the tumor suppressor gene will inactivate, so it will not control the CDK and cyclin, so it lead to cause cancer. Why? Because cyclin and CDK will not under control by the gene expression is the phenotype. So here is a proliferation of the cell will lead to cause cancer due to the uncontrolled cell division with the damaged DNA, as well as the further is the stages of the cancer will lead to cause severe process. So that means when one division abnormally will lead to more damage and the next division cause the more and more damage. So here let's begin to understand the mechanism of uncontrolled cell growth. So the oncogene is basically is contained of increase the oncogene and RAS gene, MYC gene is the oncogene increase, and tumor suppressor gene P553 and APC and BRCA is 1 and 2 will be decreased. So that means cause the cancer. So how that mechanism will be produced, let's begin to understand. So the inhibition of cell driver protein should be, but here is not because the tumor suppressor gene. So let's begin to understand the oncogene activation. When the mutation will cause, this mutation is basically this mutation when oncogene is the RAS gene basically activate due to the mutation, or otherwise it is inactive. So remember about that. When activation of this RAS gene due to the mutation or point mutation as well as MYC gene activation due to the acetylation or something which that is a genetic modification to basically lead to it is normally work basically lead to it is activate, but when it is inactivate, so it lead to increase the growth factor. So here is we'll go into the transcription to lead the RAS protein. This is the growth factor are present in the outside, so it is lead to activate to cascade of intracellular in phosphorylation. So this phosphorylation intracellularly will lead to transcriptional factor which that is transcribe the cell growth protein for G1 phase and S phase. So the CDK which we bind with the cyclin to our production. Why? Because it is increase the growth factor and it is basically is due to the mutation. Why mutation? Because the tumor suppressor gene is inactive which that is controlled. So let's begin to understand. First of all, this oncogene is active all time, but when the tumor suppressor gene is inactive, so that is why the cancer will cause, otherwise not. So here is the cell growth protein for G1 and S phase. CDK and cyclin will lead to our production, and on the other hand the MYC gene is also activated by this other mutation to not other mutation is basically is normally, so it is produce the cell survival, cell growth and cell activity by MYC gene. But important thing is that when DNA will be damaged of the cell will lead to a tumor suppressor gene not work. If here is the tumor suppressor gene which contain a P53 and P53 will produce the P53 gene produce the P53 protein. This P53 protein will bind with the phosphate group is known as the transcriptional factor. So the phosphorylation will be occur. So after the phosphorylation, so these phosphate attach with the P53 bind with the DNA. After the binding with the DNA here, binding with the DNA to produce the protein. This protein is after the transcription to translation to produce the protein for cell arrest. Why? Because DNA is damaged. So we should arrest that cell and not use anymore. So the P21 protein will be produced, and this P21 will inhibit the protein which that is growth containing protein. We will discuss also. So here the cell repair DNA repairs or cell repair or DNA repair after the damage is also produced from the tumor suppressor gene. So that means tumor suppressor gene if inactivate due to the methylation and epigenetics or point mutation will lead to cause cancer because the cell growth protein and cell survival, cell growth, cell activity by MYC gene, which that is basically is remain active and not inhibited by the tumor suppressor gene. So let's begin to understand how it's processed. So here the cell apoptosis will be occur. So the damaged DNA containing cell will be apoptosis and it will be lead to recycle if the P21 protein are not present due to the tumor suppressor gene inactivation, so will lead to cause cancer due to the not correction of the DNA and proliferation and cell growth protein will be active. So that is the tumor suppressor gene is activate to decrease the risk of the cancer causing gene, which that is the oncogenes. So thanks for watching. Please make sure to subscribe.